Well, maybe we should go ahead and begin. I'd like to good morning, and welcome everybody to the first ever Speaking of Oats webinar. My name is Tom Rabe. I work for General Mills. I'm the chair of the board of Oat Global. And Oat Global is a public-private partnership committed to improving resilience and quality and value of oats by coordinating pre-competitive research, breeding, communications, and extension on a global scale. This webinar series was a vision that came out of several of our recent Oat Global board meetings. And it stemmed from the fact that due to all the travel restrictions around COVID and our inability as the oat community to get out to see our colleagues in person via conferences and field days, farmer events, grower events, or other collaborative meetings, we decided to put our heads together and try to come up with a way to put together webinars to keep this community together and talking. We quickly partnered with the Oat newsletter. Charlene White, the editor of the Oat newsletter, who waved there, who's garnered a lot of years of resources and contacts and credibility with this Oat community. We knew that the Oat community widely looks to and follows that communication stream from the newsletter, and so we felt that that was an appropriate way to get this communication out there. So I want to thank both Dr. James Bradeen of the University of Minnesota, who is very instrumental in leading our Oat Global effort, and Charlene White of Agriculture and Agri-Food Canada for all the support and organization that she has done on bringing this webinar series forward. So speaking of oats, will happen about once a month and should cover a wide range of topics, everything from breeding and genetics of what we're going to hear today. We're going to have some discussions around the milling, from the milling industry. We hope that maybe someday we can talk about other agronomy topics, grower topics, and just general topics in nature overall. And one thing we're asking for from the participants that are on this call are volunteers and suggestions to help us put together future webinars, sessions, panel discussions, and other ways of communication for these webinars. So all are welcome to participate in that, and please send your comments to Charlene or myself or the University of Minnesota's Oat Global Jim Bradeen website. Before I turn it over to our guest speakers today, I want to let everyone know that we'll be using the chat function within the Zoom call today for questions and for communication answers, or if you're having some other technical difficulties, place them in the chat section, and Charlene and Jim will manage those questions, and they will also be coordinating the overall Zoom effort today. Today's topic is one that I've been following for the last several months, so I'm really quite curious to hear about it today. Why the PanOat genome? So why does the world need 30 oat genomes? The topic is going to be presented by Dr. Nick Tinker from Agriculture and Agri-Food Canada, whom I'm sure most of you on this call know, Martin Mascher from IPK in Germany, and also Jason Fiedler from the USDA ARS Fargo, North Dakota location located here in the United States. So we've got, for our Oat Global topic, we've hit at least on three different areas. With that, I am going to turn it over to Nick. I think you're going to coordinate some of the next steps and introductions.
Yes, greetings everyone. I'm just going to share my screen here to get going. And you should all be seeing my screen now, and one of the panelists will yell at me if something goes wrong. So yes, greetings. In a separate parallel universe, today would have been the last day of the International Oat Conference in Perth, Australia. And we would be socializing together after a lovely sunny field tour under the Southern Cross. So let's just pretend, and we'll try and make this event as social as it can be under these circumstances. Now, 4 years ago, when the last International Oat Conference was held in St. Petersburg, if you had told us that we would be sequencing 30 oat genomes and doing a pangenome in oat, we probably would have laughed. At that time, the real cost to fully sequence and fully analyze a single oat genome was close to $1 million, and the oat community would have been happy just to have the sequence of just one oat genome, which in fact we do right now. So why? Um, okay, then switching slides now. Again, my name is Nick Tinker, and many of you already know me because I've been working with the community for at least 25 years. And I'll be starting this session by explaining why one oat genome is not enough and introducing you to the concept of a pangenome. Then following that, the other two speakers will be going into much better detail than I will. Martin Mashu will be sharing his experience in developing pangenomes in barley and wheat. And Dr. Jason Fiedler will be telling you more about the details of how we're going to do this and how the project came together. So both Martin and Jason are relatively new to the oat community, but you will see from their presentations why we are lucky that they have joined the oat community. I'm going to segue into Martin's introduction at the end of my talk. And for those of you who don't know Jason, he is partly our new Xiaomin Shou running the genotyping lab in Fargo. But Jason also brings to the oat community a wealth of knowledge in translating statistical genomics and molecular tools into genomics applications. So, because this is a diverse audience, just turning my laser pointer on here, because this is such a diverse audience, I want to start by putting into perspective the complexity of the oat genome. Of course, we all know that DNA is really a long submicroscopic string of subunits that are called bases. And also, you probably know that there's 4 different types of bases, T, A, C, and G, which, depending on their order, code for the genes and the proteins that make oat what it is. But let's pretend for a few minutes that these bases were just barely large enough to see with the naked eye. Say if each one was, for the US audience, 1/8 of an inch long, or for the rest of the world, 3 millimeters long. So just barely large enough to see these molecules. There we go with a scale. On that scale, if we laid out the 21 pairs of oat chromosomes from a single oat cell, end to end, they would completely encircle the globe, and oat would be truly oat global. On that same scale, a single pair of the 21 oat chromosomes would stretch from where I sit here in Ottawa all the way to somewhere in the middle of South Dakota. And as that chromosome passed through the University of Minnesota, where this meeting is being hosted, a typical oat gene would be not much bigger than a typical car. So besides the fact that relative sizes are easier to think about on a macro scale than on a micro scale, I have used this metaphor for 2 different reasons. First, a journey along the oat chromosome, along the whole oat genome, would pass through many genes and other interesting features as it went around in the world. And this puts into perspective the difficulty of finding what you are looking for in a genome. Imagine trying to find where you left your single car somewhere between Ottawa and South Dakota, if you had lost it perhaps. But on your journey along that chromosome, you would also pass through many boring and repetitive places, perhaps Lake Michigan. It's actually the boring and repetitive places that make sequencing a genome the size of oat exceptionally challenging. So secondly, the other reason for this metaphor is if we varied that route, say between Ottawa and South Dakota, just a little bit along one chromosome, the scenery might change drastically. You might not just miss your own car that you've lost, you might miss the entire city of Minneapolis. So metaphorically, one oat genome is one random route around the world, and the pangenome is all possible routes around the world. So if a pangenome simply means all genomes, how on earth do we do that? Of course, it is not, we can't sequence every possible oat genome. There's literally an infinite number of those. What we need to do is find a sample to find out where that variation is in the oat genes. And Jason will be talking a little bit about how we made that sample. So just again, to get your head around this, this is a figure from a recent review on pangenomes, and it shows the concept quite nicely. So here we see the conceptual genes from 3 different oat varieties, or a little part of those genomes rather, and different genes are colored in 4 different colors. And I hope you can see that 2 of these genes are always present, whereas the yellow and the purple and the blue genes are sometimes or rarely present. So from this, we have the concept of a core genome that's always present and probably always necessary versus a dispensable genome, which is what we call accessory genes or unique genes. Now, another concept that's important to get your head around is that this is very, very big data, big sets of data, and difficult to manage and difficult to analyze. So we've got this concept of storing these genomes as a graph. This is one of the most promising scientific areas of analyzing pangenomes. So the parts of a genome that don't vary much are stored as a single string of bases, whereas the parts that do vary are stored as alternate paths. And I hope you can see how this fits perfectly with our paths around the globe metaphor. So one more concept before we get going into the science, and that is to introduce the distinction between de novo sequence and resequencing. A de novo sequence is what you do when you want a sequence of a whole genome without any starting assumptions. It is a long and complicated and expensive process. Sequencing technology is evolving very rapidly, but we still can only sequence relatively small bits of a genome at a time. So what we have to do is chop up a genome, put all those bits back together like a jigsaw puzzle. Now, not only is this complicated the first time you do it, but the first time you do it, it's also very important that you analyze where the genes are and where the different parts of the genome are. So this is a lot of work, takes a lot of time, takes a lot of people and expertise. In contrast, you might have heard about the concept of resequencing, or people like to talk about the $1,000 genome, and now I think people are talking about the $100 genome in human contexts. This is a relatively simple process, but it requires the presence of an existing reference genome, because what you do is you chop up a new genome and align it to that reference genome. And you can still find small variations when you do it this way. You cannot find the big variations. And if a gene or a piece of the genome is not there, you're simply not going to find it this way, which is why you need a reference genome. And sadly, this is why developing a pangenome requires de novo sequencing. So when we talk about our 30 genomes, we are talking about this whole de novo sequencing of whole genomes from no starting assumptions. Now you might ask, where do all these new genes and variations come from? And Martin's going to talk a little bit more about this and put it into perspective, but this review from Nature Plants is a good summary of why plant genomes have so many different types of genes. There's things like whole genome duplication, tandem duplication, transposon mediated insertions, and so on. But there's two that I've circled here that are particularly relevant to note. Actually, all of these things probably happen in Oat, but these two are especially important to note. And one is whole genome duplication. Oat, as you may know, is a hexaploid. It's basically 3 oat genomes jammed together into one. And what that means is every gene has 6 different copies. So that's a lot of buffer, and that allows genes to be lost or even gained and modified quite rapidly compared to in a non-hexaploid or non-duplicated genome. And the secondly is gene integration, which is what happens when we go back to the wild germplasm, introduce a new gene such as disease resistance, and then the breeders like to mix that around into different varieties. So we've got quite a hodgepodge of different genes in our oat germplasm. So why are pangenomes important? Firstly, from pangenomes and other crops, it is emerging that many traits are actually affected by the presence or absence or copy number of different genes. And this seems to be especially true of genes that evolve rapidly, and one particular example is disease resistance genes. So you may have disease resistance not because of a change in a gene, but because of a new gene coming into place, or a gene being duplicated and taking on a new role. Secondly, our knowledge of complete genomes and genome diversity opens many doors that will never otherwise be opened. So not only can we discover elusive genes that we might not otherwise find if we sequence only one genome, we can make better use of wild germplasm because we'll understand better how those wild genomes have come together into our cultivated genome. We'll be able to do low-cost resequencing when we do know a sample of most of the genes and variants that are there. We can start genotyping. We can do new types of genotyping, perhaps by targeting markers to the hypervariable regions of a genome. And perhaps most importantly, which is why I've put it in red, because we can do better science, this has a snowball effect of attracting new talent to work on oat. In my day, I could argue that oat was an interesting place to work simply because there was so much to do. And now that argument starts to fall flat because there is so much more to do in a crop that has a good public pangenome. So more and more, attracting the best talent or writing the best research proposals depends on having the groundwork of good genomics infrastructure in place. And nowadays, good genomics infrastructure means pangenome. So I'm going to end my part of this presentation with a little sobering reality check. A few years ago, I did a meta-analysis of the genomics literature in oat, wheat, and barley, and I found that the yearly publication rate in wheat was about 30 times that in oat. And as genomics moves forward from infrastructure to application, this disparity is increasing, kind of like a pandemic, if you will. The second graph here, the first graph is real, the second graph is imaginary, but it's my imaginary index of multiplying genome size, genome complexity, interest, and opportunity. And I call this my oat vanity index. And on that index, I would say that oat is far more important than wheat and barley, and has far more, ought to have far more publications. So on that note, I'm going to let Martin Masher take over. And just by way of introduction, Martin is a bioinformatics guru. He's well known in the wheat and barley communities already. And back in the day of real conferences, if you were late for one of Martin's talks, as I was a couple of times, it was standing room only. So that gives you an idea. He's relatively new, he's definitely new to the world of oats, but he's already making quite a difference. And we're really glad to have him as a part of our oat community now. He's our secret double agent, if you will, who's here to help us catch up with wheat and barley. So over to you, Martin.
Okay. Thank you, Nick. I hope you can all see my slides. Yeah, thank you for the kind introduction and also for introducing all the concepts about genome sequencing and assembly. And yeah, I have to ask to bear with me because in the next couple of slides I will not talk about oat, but rather about wheat and barley, and present some results of the recent pangenome projects in wheat and barley. And in the first couple of slides, I just want to outline the structure of our projects, how we envisioned them, and from where we started. So we at IPK Gatersleben, we're hosting the National Gene Bank of Germany, and this is why we have a strong interest in characterizing genetic resources that are held in our gene bank. And when we want to understand the species or the diversity of a species, we start from our gene bank collection. So we did genotyping of our whole barley collection, which comprises samples from all over the world. And this allows us to select a representative set of germplasm that represents the species, the diversity of the species. So we have land races, elite material, and wild relatives. And for example, in our whole gene bank, we have 22,000 accessions. And although the prices for sequencing and assembly have decreased, we cannot sequence all 22,000. So we need to come up with smaller core sets that can be used for various approaches. And the smallest core set that is labeled here A is used for sequence assembly. And yeah, at the time we started the wheat and pangenome project, this was maybe 10 to 20 accessions. And the largest set named C here, this could really comprise thousands of samples, and these will be analyzed by short read resequencing and then aligning to a reference genome or to multiple reference genome that we construct in the pangenome project. And then there are some approaches in between that are used to basically use the short reads and the assembly together to discover structural variants and to also use these variants to link genotype and phenotype, for example, in genome-wide association studies, and also to find new classes of variants. So in the end, all these different approaches will give us a catalog of structural variants that make up the pangenome, so the core and the dispensable genome. And in the next couple of slides, I will show some concrete examples from wheat and barley, where there were two genome projects or two pangenome projects. And I should point out that these were really collaborative international projects. Because of the size of the genome, it's not possible to do this by a single group. The Barley Consortium was led by Niels Stein and myself from IPK Gatterslem, and the Wheat Consortium was led by Curtis Posniak from the University of Saskatoon. But there were very many international partners whom I cannot mention now for time constraints, but I apologize for this. And the Barley study, we started really out from what we call gene bank genomics, so really genotyping our whole gene bank collection of barley. We have 22,000 accessions. The majority of them are domesticated accessions that are shown here in the principal component analysis on the right. We also have wild accessions that are clearly distinct from the domesticated barleys. And then when we run this PCA only on the domesticated part, we get such a figure where we see clear clusters that correspond to geographic regions. We see on the left-hand plot the western barleys, and on the right-hand side the eastern barleys, and also barleys from Ethiopia are very distinct. And this population structure is also found in a structure-like analysis. And in the Pangenome project, we can now select representatives from all these colored groups as different germplasm groups. And this is what we did. So we basically placed the dots in this PCA diversity space that represent different germplasm groups. So for example, the first one, Akashi Riki, this is found here. This is a representative of naked barleys from Japan. We have some accumulation of dots here, and these are actually two-rowed malting barleys, which are very important germplasm group in barley. So there we have a kind of overrepresentation. And for all these 20 accessions, we got chromosome scale reference assemblies. And the first step that we did is we aligned these assemblies at the whole genome level. So you see here two barleys aligned, Morex, which is an American malting barley, and Barke, which is a German malting barley. And you see overall collinearity on the single chromosome, but there was one region where we see this this anti-diagonal, which is indicative of an inversion. So we have a big region, about 10 MB of the genome that are in opposite orientation in the Morex and Barke genomes. And then we did a systematic survey of these inversion events. And in most cases, these inversions were located in the non-recombining region in the central part of the chromosomes, where they really don't have or most likely don't have a strong effect. But we came across one curious event that is very big, 140 MB, more than half of a chromosome arm. And it also sits in the distal region of the chromosome where there's lots of recombination. And this inversion occurred in a very important cultivar, RGT Planet, which is among the most widely grown cultivars of barley around the world. We analyzed then two different mapping populations, one where RGT Planet is a parent. And as expected from theory, in this region where the inversion is, we saw repressed recombination. And as a control, we took a Morex by Barke mapping population with two non-inverted genotypes, and there we saw normal recombination. So this inversion basically represents a very big region, probably hundreds or more than 1,000 genes that are inherited as a same linkage block. So when you make a cross between this very successful elite line and another barley variety, you would get strange patterns of recombination that were actually observed in mapping populations. And the breeders were very interested in this, to genotype this event to see if it's present in their germplasm. And then we did a historic analysis to really find where this inversion comes from. And we could trace it back by known pedigrees to a Czech variety called Diamant or Diamond that was derived from by radiation mutagenesis from another Czech variety called Valtitsky. And we found that all of the Valtitsky samples that we have in our gene bank don't have this inversion, while it was present in many of the diamond samples. So our current hypothesis is that this big inversion arose by mutation breeding in the 1960s, and then because this diamond was a very successful variety that was used to develop many other varieties, a founder effect set in that this inversion spread across barley germplasm. So this is an example that with this pan genome we can really expand the spectrum of accessible variants. No one knew about this this very big variant before, and only with recent technologies this was possible to detect these big chromosomal inversions. Another example, this is from the Wheat 10 Genomes project, where we have chromosome scale assemblies for more than 10 wheat genomes. And here, Thomas Wicker from the University of Zurich did an analysis of the composition of the repetitive space. And he came across some very distinct regions on several wheat chromosomes, so 3D, 2B, and 2A in different wheat varieties, and where the repetitive space was very different from normal wheat. And all these events trace back to what we call alien introgressions, so chromatin segments that were transferred from other Triticum species into wheat. So for example, this 3D, it traces back to Triticum or Cyanopyrum ponticum. And in a large-scale genotyping study at IPK, we got genotypic profiles for more than 8,000 wheat lines, and 1,000 of them were sequenced with whole genome shotgun sequencing. And by mapping the data to the different wheat pangenome assemblies, we could actually trace or come up with allele frequency trajectories of these alien introgressions. We see, for example, that the 2A event that is present in the Jagger genome has increased in frequency in recent years, and this is likely due to the fact that this introgression carries several resistance genes to, for example, rust diseases, and this is why it was favored by breeders. On the other hand, the 1B introgression event that comes from rye also increases resistance, but it also affects quality traits. So this is why it has decreased in frequency in recent years. On the other hand, we also found some other events that were not in the Wheat 10 genomes assemblies. So for example, the 4A event, and we don't really know what it is, but it has all the signatures of an alien introgression. So even with these 10 genomes in wheat, we still cannot capture all the possible alien introgressions that are present in current elite lines. And yeah, I just want to end with a synthesis of what we found. So you probably all know this plot that represents a domestication and crop improvement as a unidirectional process that involves a loss of genetic diversity that may be irrevocable. But actually, from our pangenome studies, it becomes clear that there were also new events that arose only in the domesticated types, some of them very recently. We have also examples of inversions that arose early after domestication and then are specific to certain germplasm groups. But yeah, the most striking thing is probably this prevalence of alien introgressions in modern elite cultivars that really have increased the diversity in these cultivars. They brought in probably hundreds of new genes from alien species that are not crossable with wheat, and this was only possible through the directed effort of breeders, and by their selection, these alien introgressions also increased in frequency, so that, for example, current elite lines in Germany may harbor 5 or 6 alien introgressions. Yeah, so this was just to show what is maybe achievable in a pangenome project. I expect that many of these events, like inversions or pangenome or inversions or alien introgressions, are also occurring in oat and avena species. And yeah, so this is now the segue into PanOat. So I've modified we have modified the schema for the pangenome project by replacing the spikes with the oat panicle. But all these methods that we have developed for wheat and oat would also work very well in PanOat, and we can probably learn many new things. And with this, I'm at the end of my slides, and I hand over to Jason to present our work plan for the PanOat project.
Thank you, Martin. So I will get this going. All right, so how are we actually going to do all this? In order to generate an oat pangenome, we've assembled a global team of more than 60 members, 30 institutions on 4 continents. This is actually a relatively easy endeavor, as the oat community has always been very collaborative and pretty tight-knit. Within the group, we have folks with decades of experience in oat and genetics, genomics, plus people like Martin with expertise in assembly and annotation of genome and a lot of other crops. So the first thing we needed to decide to do was, how are we going to pick the specific lines that would be included in the pangenome? For this, we were lucky enough to have a database of genotyping information all in one place that we could use to plot out the genetic diversity. The genome assembly steps are actually being mostly self-funded by all of our partners. And so we use these graphs to pick out the lines that would represent sort of the distinct subclusters of the global panel, but also be important for the actual funders. So to provide some context, the production of reference quality genomes has traditionally been a very lofty goal. The first human genome project initiated this entire field of research and cost an enormous amount of money. The wheat genome is about 5 times larger than the human and took about the same amount of time to get, but at a fraction of the cost of the human. Oat is about 2/3 the size of wheat, and the first wheat genome was assembled about 2 years ago. Again, at a fraction of the cost, but still quite a bit. What we're seeing over the last 30 years is a dramatic improvement in the sequencing technology and capacity and time needed to produce a full high-quality sequence. Now, as Nick mentioned, a single genome is only one route along the path. And the pangenome projects in many species are already underway. Martin already talked about the pangenomes in wheat and barley that have 10 and 20 members. The human pangenome project, which just started, is going to have 350 members when it's done. So with PanOat, with the pangenome, we're generating de novo genome sequences of 30 oat varieties and species, which will each cost about 15% of the first one. So how are we able to do this? In the last 2 years alone, a lot of new technology has emerged that's going to allow us to do this so efficiently. The most important is the release of the Sequel2 instrument from PacBio. This generates high accuracy sequences that are about 15,000 bases long, or if we want to use Nick's analogy from before, about half the length of a football field. Then we can use, again, new optimized software for these long reads and huge HPC resources available in a number of different institutions to put together these sequences into things we call contigs, which can be up to about 40 million bases long, or 80 miles in length, okay? So that's a large chunk of contiguous sequence, but for an oat genome, we still need to stitch together about 300 of these to be able to generate the full chromosomal sequence. So to do this, there are several new techniques that can be performed to identify which contig sequences are next to each other in the genome, paired with, again, new software that works better than before, and a lot of expertise in how to handle all this data. Sequences that span the full chromosome can be generated rapidly now. So prior to the release of the Sequel2, we had to either start with long reads that were inaccurate or short reads that were only a few hundred bases long. And in both cases, assembling that into full chromosomes was a lot more difficult. So the pipeline's working pretty well so far. Each box here is an alignment of individual chromosomes of 2 different genomes that the community just recently generated. The strong diagonals here show that there are very few, that there's very good collinearity between these genomes, and these little boxes show some structural variation of inversions. Now, we don't expect such good collinearity in the entire pangenome, especially for some of the more distantly related varieties. So one important part of this pangenome is the characterization of the transcriptome, or all the genes. The PanOat project has planned out this effort right from the start because in many ways, annotating is more difficult than generating the genome sequence itself. So how are we planning to do this? Conceptually, it's actually pretty straightforward. Genes in a DNA genome are transcribed to RNA, and those are translated into proteins. We have kits that we can use to extract the RNA and prep it for sequencing. With the new PacBio instrument that I just mentioned, we can sequence all those genes, one shot, one flow cell, head to tail. Practically speaking, RNA is really unstable and is mostly transiently expressed depending on a myriad of factors such as tissue type, developmental stage, time of the day, stress, disease, a whole bunch of factors. So you need to be very careful with the proper pooling and replication to make sure the extracted RNA captures a representative sample of the entire transcriptome. Ideally, we want to get RNA from every single gene and gene variant all at one time. So it's very challenging logistically. And we're trying to do this simultaneously on 24 lines that have been selected for their diversity on a global scale. So this image here shows the panicle of some of the lines about 30 days after planting, and you see quite a range of developmental stages at a single time point. So in order to do this well, we needed a single location to perform all the RNA work and expertise in these techniques. And we were able to find this at the Natural Resources Institute of Finland, Luka, where Lydia Bitz has been managing and performing this work since the spring and been doing just a phenomenal job. So Luka and USDA had never worked before together, and so it took a little bit of effort to get the agreements established to fund this work. But I think this is going to be a start to a very productive collaboration between these two institutions. I should also note here that a lot of the sequencing for PanOat is occurring at Brigham Young University, including all of the IsoSeq libraries being generated in Finland. They just purchased a second Sequel II instrument, which really helps speed up this entire process. So once we have the sequences of the genes from all of our representative tissues, the data will be sent to Manuel Spenningel's team in Helmholtz to perform the annotation. And they're going to use a combination of genome alignment, prediction, and comparative knowledge in related species to pick out sort of where the genes are in each of the genomes. Now, in addition to flagging where the genes are in the genome, we also want to draw some information on how these genes are expressed. Another reason to do all the RNA work in Finland was to minimize the environmental variation. Our plan is to measure RNA abundance in 6 tissue types of the 24 lines that we're annotating. Then we can look at differences such as during flowering, what does gene expression look like in, say, line 1 versus line 2? Now, it's surely not a comprehensive expression atlas, but it provides a really nice foundation for subsequent studies to build off of, which is the whole point of this pangenome. So here's a rough outline of our pipeline of what we're doing here. So we've already picked out all the germplasm and made a mad dash to distribute seed to all the multiple parties to perform a lot of the work in parallel. So sequencing is underway at various service providers. Hi-C is occurring either at IPK in Neil Stein's group or with some commercial providers. And then Lydia in Luka is finishing up the growing season in Finland. So some of the RNA will be sequenced at Luka and some will be sent to BYU for PacBio sequencing. Once the raw data is all generated, Martin and Manuel will take over all the bioinformatics of assembly and annotation. And once we have all of that, PanOat has several working groups we've already planned out that will focus on various aspects of the analysis with the final goal of releasing this all to the public. In general, most of the lines are following the exact same protocol for assembly, but there are a few differences. For example, some of the lines have already been assembled with ultra-long nanopore reads, and some are going to be assembled actually starting from the short read data, but with an optimized approach that Martin developed. So here is our timeline. PanOat had our first official meeting about a year ago with a large in-person meeting at PEG right before the pandemic hit. Nonetheless, we're sort of halfway through our project, and it's going to take about another half a year or so to complete the data generation step and the assemblies. And annotations hopefully will be done in September of 2021. Ideally, we'll all be able to get together in Perth a year from now and present some findings. And then after that, we'll get the data out to the partners, and then analysis will occur next year with hopefully a publication in 2022. So finally, how are we planning to use this thing? Well, with a large group with diverse interests and areas of expertise, there's a lot of information to be gleaned from a pangenome. For example, some of us will focus on basic genomics questions such as evolution and domestication and hybridization. Others were more interested in characterizing and categorizing the genes into core dispensable units. We'll also use the pangenome to assist in ongoing mapping efforts to find causal genes, identify markers that influence traits. With improved markers, we can actually enable breeders that want to use marker-assisted selection and eventually genomic selection in their programs. But overall, this resource will enable us to ask a lot more questions than we could before. And again, it's going to provide a really nice foundation for a lot of new research moving forward. So with that, I just want to wrap up with acknowledging the PanOat contributors. I won't go through all the names here, but I want to acknowledge Martin and Nick, who are kind of our co-leads of the project. I also want to acknowledge our partners in China and Australia, who are both contributing multiple genome sequences and have to attend conference calls at really awkward time slots. Finally, I want to acknowledge Lydia, who again has been carrying a lot of the PanOat's water this growing season with all of the RNA work that she's been undertaking. And with that, I will wrap it up and thank you for your time, and let's open it up for questions if we can. So I need to stop sharing my screen.
So there was one question I think people can see there from Pat Heslop-Harrison. He was wondering whether a preprint of that Cody et al. Paper was available yet. And no, apparently the manuscript is still in press. Perhaps at some point, Martin can let us all know when it's available. Also, I did see that Yuanying Peng had her hand up. Yuanying, did you want to put a question in the Q&A box, or Grant, is there a way we can let her have the floor?
Yeah, if you could raise your hand again for me and I can bring you up.
Just gave access. You should be able to ask your question now. I think she's gone.
Okay. Well, there are a couple other questions.
She's in the panelist right now, so she should be fine. If she can unmute and she can ask a question.
Well, while we're sorting that out, there are two other questions as well. Axel would like to know, is there a plan to grow out the 30 pure lines of oats for the pangenome to thoroughly characterize the phenotypic variation? Would anyone like to tackle that?
Nick, you're muted.
Yeah, so excellent question, Axel. Can you all hear me?
Mm-hmm.
One of the points, as you know, Axel, is that we want to be able to distribute this seed for other work that may be done by others outside of the community, especially once these start to become world famous lines. There's probably be a lot of wealth of information that could be gleaned from phenotypes that we can't see in Finland, and phenotypes that you might be able to see in different locations. So the plan is that right now all of those lines are, the pure lines are in Finland and Germany, because that's where the DNA work and the RNA work is being done. So we're planning actually for you to receive the lines, Axel. So in the long run, people will be coming to the Canadian Gene Bank to access that germplasm and hopefully grow it out and do other things with it. We don't have, as part of this scheme, a plan to grow those in multiple locations, but that's actually something we might start thinking about sooner rather than later. So thanks for that question.
I want to elaborate a little bit that in December, we will hear from folks about the International Oat Nursery as part of the Speaking of Oats series. So there might be opportunity to leverage that resources to accomplish some of the phenotypic testing that's being proposed.
Sorry, I was muted there. Jeff Mon would like to know, why not do more wild relative genomes?
Shall I answer that? We are doing, it seems that things are getting added on that may or may not be part of the project. One of the issues is that we're trying to do these things in parallel so that we can do the RNA-seq. So something that gets added on may be sort of a sub-project or maybe part of PanOat too, perhaps as the barley community is doing, and maybe Martin wants to say something about this. There'll be a second generation of pangenomics that concentrates on different germplasm, such as the wild oat germplasm. But the cost of sequencing is coming down so quickly that many of us are already thinking about projects to sequence wild relatives, because there's a really fascinating amount of information that can be gleaned from where the genomes of oat came from. And you're going to see that emerge from the small number of wild relatives that are included already. But I think it'll just snowball.
Yeah, I can add that the pangenome is not a fixed project. So, but I think pangenomics will be the future, and probably maybe in 5 years we will speak about 1,000 oat assemblies if the technology is progressing so fast. So then there would be actually no reason not to sequence more Avena relatives.
Okay, and another question from Pat Heslop-Harrison here. For detecting inversions and translocations, do you plan to use high-C on all the accessions, or use deep sequencing to find the breakpoints, or in situ hybridization with oligopools?
Yeah, I can cover this. So we do plan to do high-C on all the 30 accessions to construct chromosome scale pseudomolecules that will be validated by the high-C contact matrices. And yeah, when we see then inversions or translocations in the assembly alignments, we have the independent validation by high-C. And yeah, with in situ hybridization of oligopools, this would be an additional validation. We could think, for example, for complex regions like centromeres. But I think the first step is to do the high-C.
Okay. Mark Sorrells would like to know what IP restrictions there might be. So intellectual property restrictions.
Mark, that's a good question. It depends on the line, I would think. Many of these lines that we're sequencing are cultivars. Some of them are available in GRIN right now. So I think we would have to go through the individual contributors to get access to that.
So Mark, I'm not sure if you're referring to the physical seed or to the genomes or both. The physical seed, I think there's been a consensus on the call so far that everyone wants to make their lines available for further work. I don't know of any that won't be. In terms of when the data from individual genomes will be released, that's actually going to be flexible. Obviously, they're all going to be released on publication, but we're working on a contractual agreement now. Actually, it's with the IPK lawyers to get started. And that's basically going to make sure that the genomes are available on an as-needed basis for those people that are doing the meta-analysis and the work where they need to see it all. So there may be an opportunity for one partner, if they can't release the genome publicly right away, to keep that amongst those doing the work. And as the project matures, I think we'll make sure that all partners have access. And we don't have a plan to make everything publicly available before the publication, but that's something that could evolve as well.
Great. Thanks. There are a couple of people, Stefan Boyk and also Mike McMullen, would like to know if one of you can give a more detailed overview of all 30 single oat genotypes, or share the list and the names in any way. And also, Mike would like to know whether there's a naked oat line in that list.
Yes, yes, I can maybe bring up a share. I'm not sure if we're going to have time to talk about all this, but I don't think there's any reason we can't share the list of the lines that's available. I'll double check with our group and see. We're now at the stage where we have an informal agreement among the collaborators of the PanOat project that we need to answer a set of questions before we join into future conference calls, and that may include getting access to this list, but let me just pop it up on my screen. So there's the list of, actually we lied, it's right now the list has 31 lines on it, but the list is there if you want to quickly memorize that. But I think what's of more relevance is where those lines are coming from. And Jason talked in his part about the fact that it's a mixture of covering the diversity and making sure lines, the people that are paying for lines to be sequenced have things that are of interest to their country there. Gail is one of the naked lines to answer that question, but the Chinese group under Yan Peng is also contributing their data from two naked oat lines, both a cultivar and a landrace, I believe. So we'll have good coverage of naked oat, and I'm sure if you picked apart this list, there would be things in there that you could point at that we're missing. But the important thing is that we have covered all corners of that principal component analysis to some degree. Tim Langdon, I'm not sure if he's on the call, but Tim has actually started into a lot of this work already before it was possible to sequence complete genomes, and he's got contributing several lines that cover some of the Avena sterilis, sort of the progenitor of hexaploid oat. So that's also covered. Does anyone else want to add to that?
No. So Josh Anderson, and I think, well, Wekai also had a question relating to the same thing. Is there an important trait that could be otherwise very difficult to improve in breeding, but a pangenome would make it easier?
I think I can answer this from our experiences in wheat. So with all these introgressions, without a proper genome sequence, it's very hard to see what is going on there. And in the chat, there was also genomic selection mentioned. If we, for example, do GWAS or use genomic selection models, there would be a large linkage block that is very hard to explain. But with this, with the pangenomic information, you could basically use this introgression, the presence of the introgression as a marker. And yeah, only with genome assembly will it be possible to capture genes that are in the introgression. These genes could be related to disease resistance or also to other traits that are influenced by introgressions.
Okay, thank you. And Monica Matthias, do you have information about the proportion of unique genes in comparison with core genes in the oat pangenome? Many thanks and very interesting.
You may have to take that one too, Martin. I would have guessed.
Yeah, I think this is what we will have after we have all the assemblies. So we are still in the progress of getting the assemblies. So far, we have mainly looked at structural variants. Maybe from our Barley experience, we looked at kind of a single copy pangenome, and we found that in the single copy region, it's a single copy region present in a single genome is 400 MB, but when we go to the whole 20 accessions, it's 650. So I mean, each single line is if you have just a single reference assembly, you're missing more than half of the genes that are present. And so even if we have a single reference, we may miss a lot. Yeah, but to see what are really genes that are unique or occur at very low frequency, we would need the full 30 genomes to really get to look at the allele frequencies of the presence-absence variant.
Okay, and I did see that Yuan Yang has made a comment here. that they have sequenced another A genome diploid, A damascena. So that's Yuan Yingpeng from China. Oh, I missed a question here, I think. Bindu Sunil Kumar, how could pangenomic information help to identify the mycotoxin, including DON and ochre toxin, in varieties all over the world from these data?
So I'm assuming you mean the resistance rather than the actual mycotoxin in the lines. You've picked probably the most difficult disease trait to talk about. As we know from our barley and wheat colleagues, it's a very quantitative trait. It's probably even more quantitative in oat, but compounded by the difference between naked and covered lines. So are we going to answer questions about mycotoxin and DON resistance from this? I certainly hope we will, but this is, this is where the communities are going to have to take the information and start running in new directions with it. This is really an enabling process. So as with Axel's question, we're going to need more phenotypes because we're getting to the stage where we can make oat genotypes for $1,000, but we still have to do phenotypes, good quality phenotype data in the right environment, but it'll very much depend on putting that together with the genomics.
There's another question from Whoopi, but do we have time for that?
I think we can do one more.
Okay, so for weak eyes, I actually
Want to talk about practical haplotype graphs, Jason?
Oh yeah, so so generally, the question was, is PanOat necessary for genomic selection in oat breeding? And not necessarily, but as someone who runs the current oat 6K chip, it's really underserving the community. In general, oat doesn't have a good way to do genotyping. For a long time, they were doing GBS with this kind of non-reference thing that Nick had developed. But having a reference genome and having multiple reference genomes is going to make this a lot easier to develop good markers. So in general, all of your mapping questions and trying to find genes and genes cloning, we need to have a good set of markers to start from. And so with this pangenome, we'll be able to identify unique, and Nick mentioned this a little bit, hypervariable regions, regions where one marker will suffice for hitting a haplotype or a linkage block. So we're really, the next thing to do after this pangenome is developed is to use all that information to develop a better genotyping assay or array. And that will ideally meet the requirements that are necessary for genomic selection and also meet the requirements for high density mapping, at least a first round pass of it. There's also, again, Nick mentioned the graph representation. There are ways to condense all this information down to a more usable form, and we'll be working on getting that made as part of the working groups.
We're still receiving questions, but should we answer those now or answer them later and send everyone a document? Because I see that Tony Eisenhut has also raised his hand.
I think in the interest of time, we better cut this off, but we'd encourage people to put your questions in the chat and we'll try to get those answers to you. So this will be posted on the Oat Global site as well as the Oat News site. So we'll include annotated Q&A document as well, assuming our panelists are willing to take a moment to answer those questions. So just drop your questions in the chat and we'll get those going. Tom.
Yep. So with that, then I guess thank the panelists, Nick and Martin and Jason, for a wonderful presentation today. I also want to thank everybody who attended. I saw over 110 people participating in this event today. I think that speaks volumes to what we're trying to accomplish here and communication within the oat community. I want to thank the organizers, Jim and Charlene, for setting this all up and for arranging the next few conversations we have on upcoming and for managing the process today. Don't forget to send in future ideas for future events or panels. We have a couple, 3 in the pipeline already. As mentioned, we're going to hear from the North American Milling Association a month from now, along with the Canadian Milling Association. Jim mentioned that we're going to talk about International Oat Nursery in December. So keep those in mind, and again, send in ideas. And don't forget to register. There's a link in the chat section, and you should be receiving an email to register for the next event on November 19th. And then by all means, forward and share that with others who might be working in the oat community, and very involved, that most of us are, or maybe on a periphery as well. They might find some interest in these as well. So share the link and the invite to them as well. With that, I think we're ready to wrap up, and thanks again for attending. Okay.
Bye, everybody.